Namaskar. Today we'll study the IP3 DAG pathway of G-protein coupled receptor, and this pathway is shown by GQ type G-protein coupled receptor, where the effector is phospholipase C. So there are four major families of G-protein coupled receptor. GQ type activates phospholipase C. So phospholipase C beta is a type of phospholipase C. So let us see why it is called G protein coupled receptor. G protein stands for guanine nucleotide binding protein. GDP, GTP are the guanine nucleotide binding proteins. So in the resting state, alpha is bound to GDP. Alpha, beta and gamma are the three subunits of G protein, and GDP is bound to the alpha subunit. And when there is GDP-GTP exchange, GTP binds to alpha, it becomes active, and alpha GTP gets separated from beta gamma. They will bind to specific effectors of G protein coupled receptor, which may be either enzyme or ion channel. And the enzyme may be adenylyl cyclase and phospholipase. Ion channel may be potassium or calcium. In case of GQ type G protein coupled receptor, the effector is phospholipase. So let us just see this one, how G protein coupled receptor operates. In absence of the ligand or agonist, alpha GDP is there, alpha beta gamma, and GDP is bound to alpha. When the ligand binds to the receptor, GDP GTP exchange occurs, and alpha GTP, when it becomes alpha GTP, it is activated, gets separated from beta gamma and the receptor. The G protein has two properties. One is it is highly diffusible on the cell membrane, and it can freely move on the cell membrane, and it is highly specific. So freely move, it will alpha GTP and beta gamma will freely move and will bind to specific effectors. And when they bind to the specific effectors, there will be activation of the GTPase. GTPase will hydrolyze GTP, it will again become GDP, and it will get dissociated from the effector, and beta gamma will also dissociate from its effector. Then both of them will return back to the receptor, and the resting state will come. This is the way in which G protein coupled receptor functions. This is the G protein coupled receptor cycle. And when it is GQ type G protein coupled receptor, this effector is phospholipase C, and when it is phospholipase C, the target is phosphatidyl inositol diphosphate. Phosphatidyl inositol diphosphate is located on the cell membrane, and its structure is like this. And when phospholipase C targets this phosphatidyl inositol diphosphate PIP2, it becomes two parts. One is inositol 1,4,5 triphosphate IP3, and the second part is containing fatty acid, arachidonic acid, is the diacyl glycerol. So PIP2 in presence of phospholipase C becomes IP3 and DAG. Now IP3 and DAG, we call them as the second messengers for this receptor. So when the agonist binds to GQ type of G protein coupled receptor, IP3 and DAG are formed. These are the second messengers. IP3 acts on the sarcoplasmic reticulum to release calcium. Calcium binds with a cytosolic protein called calmodulin to form calcium calmodulin complex, and calcium calmodulin complex activates protein kinases to produce the cellular effects. And similarly, diacyl glycerol in presence of calcium and phosphatidyl serine activates protein kinase C. Active protein kinase C can phosphorylate other proteins and activate them to produce the cellular effects. So let us see this through one example, and let us take the example of alpha-1 adrenergic receptor. As we know, adrenergic receptor of two types, alpha-beta, alpha-1, alpha-2. Alpha is of two types, alpha-1, alpha-2. Beta is of two types, beta-1, beta-2 and beta-3. And this alpha-1 is an example of GQ type of G protein coupled receptor. and the agonist here is noradrenaline. Noradrenaline has more affinity than adrenaline for alpha 1 adrenergic receptor. So it is a GQ type G protein coupled receptor, and alpha GTP will, here alpha Q GTP will bind to the specific effector here, and the specific effector is phospholipase C beta, which will convert PIP2 into IP3 and DAG. And here in the alpha 1 receptor is present in the blood vessels or the vascular smooth muscles, and in the vascular smooth muscles there is a protein called myosin light chain kinase, and this protein is activated. How it is activated? Calcium binds with a cytosolic protein called calmodulin to form calcium calmodulin complex, which activates myosin light chain kinase. And if you see the structure of myosin light chain kinase, myosin light chain kinase can bind to both actin and myosin, which are the two regulatory proteins of the smooth muscles, and it has site for binding of calcium calmodulin complex. And when the calcium calmodulin binds to MLCK, the catalytic site is activated, and there is also a TP binding site within the catalytic site, and this will phosphorylate the myosin. And if you see the mechanism of contraction of vascular smooth muscles, or contraction of and relaxation of smooth muscles, it consists of the thick filament, which consists of myosin, and the thin filament, which is actin. There are 10 to 15 thin filaments for each thick filament, and when these heads, myosin heads, so this thick filament consists of myosin heavy chain, and this is the myosin light chain, and this myosin light chain, when gets phosphorylated, will bind to the actin, and 10 to 15 thin filaments for each thick filament are there. So when all of them will be bound, actin myosin binding will occur, the smooth muscle will be contracted, and for that this MLCK is required, myosin light chain kinase, which phosphorylates the myosin light chains. And so myosin light chain is phosphorylated by active myosin light chain kinase, and now this myosin phosphate has the energy, it will bind with actin, and actin myosin binding results in contraction of vascular smooth muscles. Similarly, diacyl glycerol also causes contraction of vascular smooth muscle. And diacyl glycerol, as we discussed, it will activate the protein kinase C. Active protein kinase C will phosphorylate another protein called CPI17, and this CPI17 will inhibit the enzyme myosin light chain phosphatase. CPI17, why it is called? Because the full form is C kinase, protein kinase C. C kinase potentiated protein phosphatase 1 inhibitor. So it is a type of myosin phosphatase inhibitor protein, and protein kinase C phosphorylates this protein and activates this protein. Now this active CPI17 will inhibit myosin light chain phosphatase. Myosin light chain phosphatase is required for dephosphorylation of myosin phosphate. So if it will be dephosphorylated, it will again become myosin light chain. So by inhibiting myosin light chain phosphatase, there will be increase in myosin phosphate level, so there will be actin myosin binding and contraction of vascular smooth muscles occur. So IP3 and DAG, both these second messengers play a role in contraction of vascular smooth muscles. So this is one example, and in this way IP3 DAG pathway occurs in case of G protein coupled receptor. Thank you.